Hello friends, myself Dr. Kriti Pathrikar, Chief Embryologist, Samarth IVF. Today I'm back with another burning topic known as PGD or Preimplantation Genetic Diagnosis. So friends, we'll learn today why PGD or why not PGD. So first of all, we'll know what is PGD. PGD is a genetic screening of embryos before transfer for detecting out any kind of anomalies in the genes of the embryo. PGD provides a cutting edge in the cases of embryo selection where one or both of the parent is genetically predisposed for diseases, where the embryos can be selected before the transfer to rule out the passing of the genetic disease to the progeny or in cases of recurrent pregnancy loss. So now let's see why PGD. First is in the cases of monogenic disorders. Second is HLA matching, where the the resultant pregnancy will serve as the savior child. To rule out the cancer predisposition in the progeny. Also, in cases of minor disabilities, the genes which are having minor disabilities, to rule out that it doesn't pass on to the child. So now, how is this PGD done? PGD is done in two ways. First is a biopsy method, which is an invasive method. Second, nowadays it has come up to few non-invasive methods. Let's see one by one. First are the invasive method, that is a biopsy technique, in which first is a polar body biopsy. In polar body biopsy, the polar body from the oocyte is extracted from the perivitelline space and given for the testing for genetic predispositions of the embryo, and then embryos are selected on the basis of the subsequent result of the genetic diagnosis. The second biopsy method is the cleavage cell stage, where the cell from the 2, 4, 6 cell stage, the one cell is extracted. The third method is on the blastocyst stage. We can also have a biopsy small section. And the last method is the cumulus cell biopsy. In all these 4-5 types of biopsy techniques, the most prevalent ones are the cleavage cell state biopsy or the blastocyst biopsy, because those are the most sturdy ones. In cases of cumulus cell biopsies and the oocyte polar body biopsy, we can only rule out the maternal side of the genes but not the paternal side. So the most prevalent used methods in cases where the male factor is involved for the genetic predisposition of diseases, we will use the cleavage cell stage or the blast stage for the biopsy. Let's go on to the non-invasive methods, which are the preimplantation screening methods, where the blastocele fluid is either used for the screening of the DNA, or we use the fluid where we are keeping the blast. Now let's come up to the major disadvantage of the PGD, which is PGD while doing the process or after doing the process can result into 20% of embryo loss. That is why even if it is a very modern technique, most sought after technique, still we'll avoid doing PGD in cases where it is not indicated. To see more information on infertility related topics, please watch our channel Samarth IVF. Thank you.